Welcome to Cancer Lesson 2.5, How is Cell Death Controlled in Normal Cells and Disrupted in Cancer? The goals of this lesson are to explore how controlled cell death, called apoptosis, is necessary for the health of an organism, and consider how lack of apoptosis can lead to unregulated cell growth. At the end of the lesson, the students should be able to explain how controlled cell death, or apoptosis, is an essential part of the dynamic equilibrium that exists for all cells. Describe how cell survival is a dynamic process that requires extracellular signals. And explain how the balance between apoptosis and survival is shifted in cancer cells. We will achieve these goals by engaging in a Socratic discussion and a card game about apoptosis. To prepare for this lesson, you'll need to review the following key scientific concepts that will be presented throughout the lesson. In healthy tissue, the balance between cell division and cell death is precisely controlled. Controlled cell death, called apoptosis, is initiated by dye signals that counteract the survival signals that regulate cell division. Dye signals arrest the cell cycle, giving the cell opportunity to assess whether DNA is too damaged to repair. If it is, the cell undergoes apoptosis. The gene p53 is the master regulator of apoptosis. You can review the scientific content in the background reading provided for you in the teacher primer, the teacher manual, and the student workbook. The teacher primer provides in-depth knowledge about the scientific content presented in this lesson. The teacher manual, or lesson plan, provides a minute-by-minute explanation of the lesson structure. The student workbook provides additional explanation for students. Be sure to print the lesson worksheet, the homework worksheet, and the apoptosis card game. The key points of the do now are that cells die by necrosis as a result of random external damage. Cells die by apoptosis as a result of dye signals that activate the p53 gene. p53 arrests the cell cycle, so the cell can assess the extent of DNA damage and decide how to respond. When a cell is no longer functional, the cell chooses to destroy itself in a process called apoptosis. But there are two ways cells can die: necrosis and apoptosis. Necrosis occurs when a cell is physically injured, such as when it is infected by a virus. In this case, the cell explodes. Apoptosis occurs when a cell receives signals that it needs to die. These signals may come from the outside or from the inside of the cell. Both external and internal dye signals activate a gene called p53. p53 is called the master regulator of apoptosis. p53 stops the cell cycle at the restriction point, which is at the end of the G1 phase. Once the cell cycle is stopped, the cell assesses the extent of DNA damage. If the DNA can be repaired, then the DNA will be repaired and the cell will survive. But if the DNA is too damaged to be repaired, then the cell will go through apoptosis. p53 is called the guardian of the genome. The key points of the discussion are that p53 initiates apoptosis by opening pores in mitochondria. Enzymes called caspases then leak out of the mitochondria into the cell and chew up cellular proteins and DNA, killing the cell. Survival signals block the mitochondrial pores, preventing them from leaking caspases. How does apoptosis happen? Dye signals from either outside or inside the cell activate the p53 gene, which encodes for a protein called p53. p53 stops the cell cycle at the restriction point, so the cell can assess the extent of DNA damage. If the cell's DNA is too damaged to repair, p53 signals the mitochondria to release enzymes. These enzymes, called caspases, leak into the cytoplasm where they can chew up the cell's organelles, membranes, and DNA. Note that once mitochondria pores open to release the caspases, the cell is at a point of no return. It must kill itself. As the caspases chew up the cell structures, the cell changes its shape. Eventually, the nucleus fragments. Once this happens, the cell is dead. The cell parts are engulfed by other cells and recycled. Apoptosis is part of the dynamic equilibrium between growth and death that maintains the health of the tissue. In fact, signals for apoptosis are always present. They exist in equilibrium with survival signals. When survival signals prevent die signals from working, the cell will survive. When die signals are not opposed, the cell will initiate apoptosis. How do survival signals counter apoptosis signals? Survival signals tell the cell to make proteins that block the mitochondrial pores. This prevents caspases leaking into the cytoplasm, initiating apoptosis. Consequently, the cell does not die. Signals that control cell fate work in the same fundamental way as signals that regulate the cell cycle. Note that this slide is animated to show each step. First, external signals activate their receptors on the cell surface. Next, receptors activate transduction proteins inside the cytoplasm. Transduction proteins in turn tell transcription factors to move into the nucleus. If the gene is in an uncoiled state, the transcription factor can attach to it and initiate transcription. If the gene is p53, it controls either cell division, if damaged DNA can be repaired, or apoptosis, if it can't be repaired. For the lesson activity, students will play a card game that is similar to card games like Gin or Rummy. The purpose of the game is to simulate the dynamic activation of pro-survival and pro-apoptotic signals within a cell. Instructions for playing this game can be found in the Lesson 2.5 activity instructions for teachers and students. The card game cards for printing can be found in the PowerPoint called Lesson 2.5 Activity Cards. The key points of the wrap-up are that p53 regulates the balance between cell division and death required to maintain health. If p53 is mutated, cells with damaged DNA can carry on dividing, which may lead to hyperproliferation. Mutations in p53 are common in several types of cancer. The main idea is that some signals promote apoptosis and other signals promote survival. Tissue health requires that there is a dynamic balance between these signals. Students should recall from Unit 1 that DNA mutations occur every time cells replicate. They should also recall that carcinogens can promote DNA mutations directly or indirectly. However DNA is damaged, p53 will stop the cell from continuing through the cycle while it assesses the extent of DNA damage. If the DNA damage is small, or if it can be repaired, the cell will progress through the cell cycle and divide. If DNA damage is extensive, p53 will stimulate the mitochondrial pores to open and release caspases, starting apoptosis. But what if p53 itself is mutated? Then the cell cycle cannot be arrested. Extensive DNA damage will not be repaired, and the cell will not start apoptosis. We frequently see mutations to p53 in cancer. For homework, have students read a short article about the role of p21 in spontaneous healing and its possible use in limb regeneration and preventing cancer. One of the most common questions students ask is survival the same as proliferation? The distinction between survival and proliferation is important because many cells are perfectly healthy but cannot divide, such as neurons. Survival means that the cell is actively countering default death signals from the environment with its own survival signals. Proliferation means cells are receiving signals from the environment to divide. Another question students ask is p53 also a hub like RB? Exactly. In fact, more than 80% of cancers have mutations in p53. At the end of the lesson, collect the lesson worksheets to assess students' explanations of how signaling pathways work and their strategies they found useful in winning the apoptosis card game. In this lesson, we examine how normal cells behave as part of a tissue community. In the next unit, we will focus on those elements of a normal cell's behavior that are disrupted in cancer. Don't forget, if you have any questions, concerns, or feedback, to let us know. You can contact any of the CTSE team members and we'll be happy to help you.